bite of that egg, aren't you the least bit interested in knowing something about it first?
No.
Well, you're in luck.
Who are you?
That doesn't matter. We're here to shed some light on that egg and to show you how you and I were not so different from it to begin with.
Well, I don't really care. I just...
No nonsense. A little brain power right before breakfast will go a long way. We'll give a new meaning to the phrase, you are what you eat.
Where are we? Why is it so dark?
Never fear. We're looking right now at where it all began.
Where what began?
Life, of course. Complex organisms such as you and me all started out as a single cell, or should I say as two halves of a whole cell. When mommy and daddy decided they wanted to have a baby, they facilitated a process called fertilization that resulted in a zygote, a single cell with the potential to become a complex organism. Now, all of this begs the question, how did we go from this to this? The development of multicellular organisms from a single cell, the fertilized egg, is a brilliant triumph of evolution. During embryonic development, the egg divides to give rise to many millions of cells, which form structures as complex and varied as eyes, arms, the heart, and the brain. This amazing achievement raises a multitude of questions. How do the cells arising from division of the fertilized egg become different from each other? How do they become organized into structures such as limbs and brains? Much of the excitement in developmental biology today comes from a growing understanding of how genes direct these developmental processes.
That's really interesting. But what really happens during development? And how does a cell know that it's supposed to do all those things?
Well, I'm glad you asked, Lily. And that's why we're here in the lab of Dr. von Liebenstock.
What? We're in the lab?
Oh yeah, the lab of Dr. von Liebenstock and Dr. Strange. Maybe they can answer your question.
We certainly can.
But before we delve into that topic, let me first try and define it. Development, essentially, is the emergence of complex organized structures from an initial population of very simple cells. There are 5 main developmental processes. There's cell division and proliferation, and then there's body access and pattern formation. There's also specialization and differentiation, morphogenesis, and finally, growth. It's important to note, however, that I conveniently distinguish these 5 processes, but in reality, these processes overlap with and interact with each other considerably. The first of these processes is cell division and proliferation. Proliferation is the net increase in the cell number between each division and reflects the balance between cell division and cell death. Cell division and proliferation is important in the creation of multicellular organisms, which start from single-celled zygotes and become composed of millions or even trillions of cells. As we know, these cells don't form an amorphous blob. They have a distinct arrangement of parts and activities ordered along space and time. Pattern formation is the process by which the spatial and temporal pattern of cell activities are established within the embryo, which will ultimately form well-ordered structures. Pattern formation lets cells know to form what structures in which place of the body. It involves laying down the overall body plan, defining the main axes of the embryo. This specifies the anterior and posterior ends and the dorsal and ventral sides of the body. In addition, in animal embryos, pattern formation also involves allocation of cells to the different germ layers, that is, the ectoderm, the mesoderm, and the endoderm. Eventually, these cells differentiate to the specific cell identities that we have, and at some point, cells in each layer become fated to form the specialized structures that make up the body. But all of these set the stage for the rest of the developmental processes that we'll be discussing. But my colleague here, she will be the one to discuss with you the rest of the story. Dr. Strange?
Thank you, Dr. von Liebenstock. A single cell, the fertilized egg, gives rise to at least 250 distinguishable cell types. This include the muscle cells for movement, epidermal cells for protection, neurons for relay of messages, and many more. The generation of diverse cell types is called differentiation.
Consider the difference between human arms and legs.
Both contain exactly the same types of cell: muscle, cartilage, bone, skin, and so on. Yet the pattern in which they are arranged is clearly different. Thus, we can see that pattern formation and cell differentiation are very closely interrelated. In fact, it is essentially pattern formation that makes us different from elephants and chimpanzees. I'm sure you've noticed that our differentiated cells are not randomly distributed. Rather, they are organized into intricate tissues and organs. These organs are arranged in a given way. Our nose is where it should be and not in our forehead or other parts of the body. This creation of ordered form is called morphogenesis. Finally, the fifth process is growth, which simply means increase in size. In general, there is little growth during early embryonic development. Subsequent growth can be brought about in a variety of ways. Cell multiplication and increase in cell size during interphase of cell cycle, and deposition of extracellular materials such as bone and shell. All of these 5 developmental processes, cell division and proliferation, pattern formation, differentiation, morphogenesis, and growth, are neither independent nor strictly sequential. In any developing system, there will be many twists and turns in this sequence of events. These twists and turns are all directed by our genes.
Whoa! I never knew development could be that complex.
Well, yeah, and it's just gonna get even more amazing and more complex as we go along.
Now I can't eat this egg in peace.
That was the point.
So you mean to tell me that we're just the same cell through and through from the beginning to the end?
Well, not exactly. That single cell we saw in the beginning gives rise to a multitude of cells looking and working independently and differently to give rise to one functional you. There's a concept called potency. Now, we all started out as a fertilized zygote, which is the only true totipotent cell. That is, a cell from which all types of cells can come from. Totipotent cells can become embryonic or extraembryonic cell types to construct a complete and viable organism. Next, we become the embryo with the embryonic stem cells. These are now pluripotent cells or pluripotent stem cells that can serve as a source of numerous types of differentiated cells. The pluripotent cells are the descendants of the totipotent ones. They are generally considered immortal due to their ability to self-renew over many generations. Then we become fetuses with fetal stem cells. These fetal stem cells are cell types found in the organs of the fetus and can be isolated from their blood and bone marrow. They are called multipotent cells and can differentiate into a number of cells but confined only to a closely related family.
So how did those cells suddenly become specialized cells?
Specification is a reversible commitment of a cell to its developmental program. So if, for example, I took a cell and specified it to become part of the liver, but then put it in a culture of heart cells, then it could become a cardiac cell. In contrast, determination is an irreversible commitment to a cell lineage. So if I took a cell and determined it to be a part of the liver, and put it in a culture of cardiac cells, then it must still necessarily become a liver cell.
Well, if I started out as a single cell, wouldn't that mean that my DNA is the same in all my cells?
Well, that's a yes and a no.
Oh, hi, Jean. Glad you could make it. Lily, this is Jean. She's a genetics expert from the Absolutely Wilkins University.
Thanks, Arnie. There is a concept called genetic equivalence. It just means that all cells have the same genome, therefore the same set of genes as all other somatic cells, because they all arise from just one cell.
So how is it possible that cells differ morphologically and functionally? Differential gene expression is the key to that. The cell type is dictated by the proteins a cell produces, which are then dictated by the genes a cell expresses. Red blood cells express the gene for hemoglobin, while neurons express the genes for neurotransmitters. RBCs still have the genes for neurotransmitters, while neurons still have the genes for hemoglobin. Since these genes are not expressed, they remain unused, but they can still be expressed with appropriate signals. Gene transcription is the most important regulatory mechanism for controlling expression. Transcription factors regulate the expression of genes by binding to specific sites on the DNA and recruiting RNA polymerase, the molecular machine for transcription. There are also master control genes, in which if there are loss or gain of function mutations, they can lead to the development of organs at sites where they do not usually occur. Just imagine having eyes on wings. Gene transcription can be controlled through epigenetics. These involve changes in DNA without modifying its sequence. When DNA is methylated, genes can be silenced by attracting proteins that block the transcription machinery. Modifying the histones, molecules used in packing the DNA, also changes how the DNA can be accessed for transcription. As such, DNA can have two forms: euchromatin, where gene transcription is on, or heterochromatin, where gene transcription is off.
And this phenomenon is seen all over the animal kingdom.
All over the animal kingdom?
That's right. Actually, most of what we know about our human development came from our study of model organisms. Oh, just in time. That's Dr. Ranchodas from Mumbai. He's an expert in frog development and he's here to talk to us about, well, frog development.
Hi, Arnie. Oh, you must be Lily. I heard you wanted to learn about frog development. So from frog development, we can observe these developmental processes all come together. Just how did this become this to this totally different looking organism? It all starts with the egg cell, with an uneven distribution of various molecules such as mRNAs and proteins from the mother even before fertilization. This asymmetry is seen with the egg having a dark colored top known as the animal pole with a light yolky bottom called the vegetal pole. The sperm enters the egg cell during fertilization, which kicks off the embryonic process. Now the sperm does not only provide for half of the genome, it also acts as a positional signal that sets up the axis of the embryo by specifying the dorsal ventral or the back to belly axis. After the repeated cell division during cleavage, the zygote becomes a blastula. After this, gastrulation happens. Cells start to migrate into the interior of the embryo, causing the tissue to fold inward. This folding creates the dorsal lip, which is also called the Spemann-Mangold organizer, which does not only specify the dorsal side, but it also makes the nearby ectoderm turn into neural tissue. The end product of gastrulation is the gastrula. The key process is migration, specification, and determination. Basically, you have all these cells moving around in a very organized manner, and at the end of gastrulation, you now have specified the three germ layers: the ectoderm, endoderm, and the mesoderm. After these germ layers have been specified, organogenesis begins. It starts with a notochord, or the rod of mesodermal cells telling or signaling the ectodermal cells not to become skin, but to form a tube and become the nervous system. At this stage, the embryo is a neurula. After this, neural precursor cells elongate, stretch, and fold into the embryo. It forms the neural tube, which induces the changes in the neighbor cells, thus continuing organogenesis. The mesodermal tissues next to the notochord become segmented into somites, which are the precursors of the frog's back muscles, spinal cord, and dermis. Then we fast forward to the metamorphosis. How does this tadpole become the frog? Basically, it's a concept of apoptosis and differential gene expression. A lot of changes happen to prepare the aquatic tadpole for the terrestrial existence of the frog, such as the cartilaginous skull of the tadpole being replaced by a bony skull of the frog, or the regression of the gills, or the shortening of the large intestine characteristic of tadpoles to suit the carnivorous diet of the frog. And
Oh wow! I've always wondered why tadpoles look so different from frogs when they're basically just two forms of the same animal, right?
Yeah, and actually in human development, frogs aren't the only model organisms. In fact, we also study a lot the food of the frogs.
The food?
That's right, their favorite sumptuous meal, and I'm referring of course to flies. That's why we invited Dr. Drew Sophia from the Melanogaster College of the CPG Isle University to talk to us about, well, Drosophila flies. Oh, just in time!
Hey there! I'm the Fly Woman! I love flies and I love talking about them. So right now, I'm gonna tell you about their development.
The collection of the groundbreaking works of Mr. Thomas Hunt Morgan on fruit flies and mutations that cause homeotic transformations put forth the ideas of the genetic mechanism of segmentation and the specification of segment identities. Together with the works of several scientists, we have now identified the major classes of genes involved in generating the segmental body plan of the fly. Now listen closely, because this is amazing!
First, we have the maternal effect genes. These cause cytoplasmic polarity by the differential expression of maternal genes specifying the anterior, posterior, and terminal regions by their localization or activation. For example, we have the bicoid and hunchback mRNA localized on one side, which eventually becomes the anterior portion. This produces the bicoid protein anterior morphogen gradient. On the posterior side, we have nanos and caudal mRNA. Then we have the zygotic genes, which will eventually lead to the expression of homeotic genes. But before that, let's backtrack to the maternal effect genes. The hunchback protein gradient that we see regulates what we call the gap genes, called so because mutations in these genes lead to missing large regions along the axis. These genes basically divide the anteroposterior axis into broad regions. These gap genes specify the expression of the pair rule genes, which delimit the first signs of segmentation in the embryo. This is eventually responsible for the most obvious feature of the Drosophila larvae, segmentation. This is carried over to the adult, where the segments have now their own identity, such as wings and legs. Mutations in these genes affect the alternate segments. Together with the gap genes, pair rule genes regulate the segment polarity genes, which eventually establish the final segment boundaries. These segment polarity genes specify the anterior-posterior pattern across each segment, and thus mutations in these genes upset the anterior-posterior axis, producing mirror images or tandem duplications of the anterior or posterior parts. The collective expression of the maternal genes, gap, pair rule, and segment polarity genes lead to the differential expression of the homeotic genes, the master regulators of segmental identity of the fly. These specify the unique identity of each segment. Mutations in the homeotic genes lead to homeotic transformation in the segment identity. Basically, you have structures in incorrect locations. Exhibit A: legs instead of antennae during antennapedia mutation. Exhibit B: wings instead of haltere in bithorax mutants. What's even more fascinating is that the homeotic genes have a spatial and temporal order on which they are expressed during development. The 3' to 5' arrangement of the genes in the chromosome parallels their expression pattern from the anterior to posterior axis of the genes and when they are activated.
What's more amazing is that this arrangement is conserved even in mice and humans.
Well, I have to go feed my baby flies now. So bye! I hope you learned a lot!
Oh wow! So you mean that the body parts of the fly have already been specified since the beginning?
Right, and it's so wonderfully complex that any aberrations in these gene expressions from the beginning could have dire consequences to the larvae, or in the case of humans, the embryo. So it's all just a case of us fulfilling our destinies, or basically, what our cells determine us to be. So, what's on your mind, Lily?
So it's all a game? One day, I'm a single cell, and the next, I'm a whole person? So when does it end?
Well, funny you should ask. Normally, cells don't live indefinitely. Otherwise, we'd all live forever.
Is that a bad thing?
Depends on your point of view. But anyway, cells undergo a very important process called apoptosis.
Apoptosis.
During apoptosis, our cells die in a very clean and well-regulated manner. This differs from necrosis, which is a lot messier and can cause damage to nearby cells due to inflammation. Throughout our development, apoptosis plays 4 major roles.
The first of these is the regulation of the proper formation of our body structure. An example of this would be the formation of our fingers and toes. Initially, these are connected with webbings. However, apoptosis aids in the proper shaping of our individual digits by removing the cells that make up those webbings. The second role of apoptosis is getting rid of structures that we no longer need. An example would be the differential development of males compared to females. In the early part of human development, a precursor to female reproductive organs is present in both sexes. However, this disappears in the male fetus via apoptosis to give way to male reproductive organs instead. The third role of apoptosis is the regulation of how many cells we have in our body. Just imagine, if our cells kept on dividing indefinitely, we would all be gigantic. Apoptosis keeps our cell numbers in check to make sure we are of the right shape and size. Lastly, apoptosis is important in the elimination of abnormal and dangerous cells. These include cells infected by viruses, cells with damaged DNA, and immune cells that could attack our own bodies. In a way, our cells are pretty utilitarian. They would choose to sacrifice themselves for the good of the rest of us.
So, Lily, did you enjoy your pre-breakfast lesson? Did it make you appreciate more that egg you're about to eat?
Well, I'm still a little hungry, but yes. Actually, it made me think that small changes in the process could actually cause severe defects.
Oh, that's right. That's why I invited Dr. Angela.
What? There's another person here?
Oh, of course there is. You thought this was the
Where
end?
are you people coming from?
Doesn't matter. And she's here to tell us about developmental problems that could arise from genetic problems.
Cool. Cool!
Hello there, Arnie and Lily. Okay, so when something goes wrong with any of these developmental processes, serious congenital abnormalities or birth defects may occur. They can be problems with your heart, your limbs, your gastrointestinal tract, your respiratory tract, even your senses.
Now, there are 3 general types of developmental abnormalities based on what causes them. Some are genetic. These are caused by mutagens, chemicals or agents that can cause permanent damage to your DNA, and these types can be inherited. Others are environmental or caused by teratogens, which are agents like drugs, alcohol, an imbalanced diet, or even infections that cause abnormalities after fetal exposure during pregnancy.
Oh, good thing my mom didn't drink, smoke, or do drugs when I was in her tummy.
Right. Good job, Lily's mom. Oh, but the third type of these developmental abnormalities are idiopathic, meaning their cause is quite not known yet. These are the hardest to address.
So what are examples of these abnormalities?
Good question, Lil.
One of the most common would be Down syndrome. This results from problems in cell division. It's a case of what you call aneuploidy or an abnormal copy number of certain chromosomes.
For Downs, it's the chromosome number 21, which becomes 3 instead of 2 after cell division. That's why it's also called trisomy 21. Another example would be spina bifida, and this happens when the neural tube fails to close caudally during early development, which is said to be influenced by low maternal diet of folic acid containing food.
Oh, and I've got a funny one. You know about apoptosis, right? Well, problems in this could result to limb deformities, like having webbed fingers, like a duck.
Ooh, Mr. Apoptosis guy also told me about that earlier. So what about those who have more than 5 fingers on their hands?
That's also a developmental defect. It's called polydactyly.
Weird. But also interesting. I'm really starting to love learning about development. Just when I was just supposed to eat my breakfast in peace.
It is truly cool and interesting how all of these genes work together to create a normal you, and how even just the tiniest of errors or inaccuracies in these developmental processes and their timing can result to abnormalities that may even lead to death.
Amazing! Amazing! So there really is truly an amazing design!
Oh, absolutely! That's why all of this should make us appreciate where we truly came from. You know, all this learning has made me hungry.
Me too!
Hey, the egg!
Hey, that's so